I'm Jeffrey Townsend. I'm a professor of biostatistics, but I do a lot of. I have two students in my lab currently who are computational biology and bioinformatics students. I actually have a wet lab, so unusual thing for a biostatistician. And I sort of have a pretty broad set of things that I tend to look at. I'm probably going to just give you a quick overview of the kinds of projects that happen in my lab, and then I'll talk about one topic that is maybe one of the things that we're working on a lot and that most of the lab is focused on in one way or another, although there's a lot of projects that are pretty divergent from it as well. So just speaking generally, the lab works on a lot of computational questions using genomics data. So it's really appropriate to follow Bluma, who also works on genomics data and evolution. And in particular, we're interested in sequence data, in gene expression data, and in phylogenetic data. All of these data we put together in various ways to try to better understand a variety of things. And so just to get very explicit, I'm just going to first start off with a slide that just gives some example projects so you can have something in your mind as I talk a little bit about some minor aspects of these that I'd like you to know about. So types of projects that go on in my lab include performing next generation tumor sequencing and analysis of tumor data, inferring tumor phylogenetics to analyze selection and epistasis in response to treatment on mutations of tumor suppressors and oncogenes in cancer cell lineages. So, the big questions about cancer: how does it appear? Why is it there? What happens when patients evolve resistance to cancer? And we have lots of collaborations in the medical school with patient data, et cetera, to motivate these kinds of analyses, as well as using very, very large data sets from the public sphere as well. We are evaluating the molecular causation of cancer types, including the mutagens and mutagenic processes. We also investigate gene expression, developmental and systems biology of fungal host invasion and development. There's some linkages there that I'm not going to go into today, but that are interesting. And then lastly, I actually do have another thing that I do, which is mathematical modeling of infectious disease, cancer evolution, which I already mentioned, and phylogenetic information, understanding how samples are related to each other, which is relevant to cancer, but also relevant to organismal evolution in general. So quite a wide swath of kinds of projects that are ongoing in my lab and that I'd be happy to have individuals come in and do rotations on. Let me just give you kind of more of a big overview now, which is that sequencing data has provided an enormous amount of understanding of the deep history of life. So we understand a lot, a lot of it was gathered for medical reasons, but what we get is a lot of genome sequences from many organisms. We can then use those genome sequences to understand how organisms are related and how they evolve. Now we have a really great understanding of the deep history of life. But I just want to emphasize, and evolutionary biology is usually considered a historical science where you understand stuff about the deep history of life. But most of what's focused on in my lab is using evolutionary biology to understand what's happening now and to try to help people and patients and people make useful predictions that help us now. So one example of this, which is going to be the main thing I talk about scientifically here, although I'm happy to talk about any of these topics. If you have curiosity, I'll just spontaneously talk about anything I mentioned, is I'm going to talk about this cancer effect size that we've calculated. So one of the things that we generally would like to know in, I don't have a missing slide here, but anyway, one of the things that we like to know in cancer biology is which genes are responsible for cancer and to what degree they're responsible for cancer. And a lot of really good work has been done outside of my lab on which genes, like figuring out whether there's a significant overburden of mutations in tumor populations. But one thing that sort of was missing for quite a few years and that we're contributing right now is an understanding of to what degree is each gene contributing to cancer. And this is really important because sometimes you see genes very common in tumors that are common because the baseline mutation rate is very high. So what you need to do to understand how important the mutation is to the survival and evolution of the cancer is to deconvolve the prevalence, which is how often you see a particular mutation or a mutated gene in tumor lineages, into two things: the baseline mutation rate and the degree of selection for mutation in the cancer lineage. So just to illustrate that, if you think about this little diagram on the right, you have a cell that starts in some tissue, and then mutation creates variation. Obviously, in evolutionary biology, unfavorable mutations are selected against. If you knock out an essential gene, that cell dies. Some of that variation doesn't cause problems and maybe is helpful. So reproduction and mutation occur. The favorable mutations are more likely to survive even in your cells compared to other cells. So those favorable mutations then confer a selective benefit. And then what you end up with is a distribution of cells that depends both on the degree of mutation. The more mutations of a type you get, the more of that type of genotype you'll get. And then also the more it helps those cells survive, the more you'll get of that. So there's a mixture of mutation and selection creating the distribution of variants that you see in tumors. And what we need to do is try to figure out the difference between those because they have very different implications for what therapy would do. Some of the mutation that happens a lot because it's just mutated a lot, you could develop a therapy for and it would do you no good in terms of cancer because it's not actually conferring any benefit to the cell, whereas the ones that actually have a selective benefit to the cancer lineage are the things that you actually want to target, and the selective benefit quantifies the degree to which the target is useful to develop therapies for. So how do we do this? Just to get into a little bit of the technical detail, we use a lot of big data. We use a lot of computational tools, mostly tools we develop in the lab ourselves. But many of them have dependencies, obviously, from other scientific exercises people are doing all over the world. And so what we do is we figure out the mutation rate at each site by convolving these gene-based rates, and there are methods that we use from other people that do that, from silent sites and covariates with the trinucleotide context. Again, that's another tool that we use from someone else, but putting them together allows us to understand what the mutation rate is. So just to break that down a little bit, you can take every gene in a tumor genome and understand what its mutation rate is. That is how often a mutation arises in that gene as a general rule for the entire gene, not differentiating by site. By just looking at where the gene is in the genome, by looking at its chromatin state that you just heard about from Bluma, by looking at where the origin of replication is compared to where it is, by looking at the level of gene expression. All of these are covariates with the mutation rate. And then you can use synonymous site mutations, which are presumably neutral, as sort of a predictive variable along with those covariates to enable you to get an estimate for the rate that every gene mutates in any given cancer type in the genome. So that gives you a general mutation rate, and I'll just take a moment to just look at this x-axis, which shows that mutation rate varies over 2 orders of magnitude, which means some genes are mutated 100 times the rate of others. That gives you an idea of why it's really important to incorporate an understanding of that mutation rate into any estimate of how important a gene is to causing cancer. Because it's the difference between a 1 in 100 and almost 100% of individuals having a mutation, and that's just about the mutation rate. It has nothing to do with how important it is to cancer. Okay, so then you can take each one of those and also look using the same sort of tumor sequence data at what the trinucleotide mutation rate is. That is in the context of C on one side and a T on the other. How fast does an A mutate to a G? So you can do that for all of the possible permutations. And this is just a diagram. This is for lung cancer, by the way, of what the particular upstream, downstream nucleotides are and what the rates of each of these trinucleotide mutations are. Then you can thread all of those trinucleotide mutation rates onto each of these genes that we've characterized the overall mutation rate onto. And what that gives us is a prediction for the mutation rate for every single site in every gene. And then we can play that through the genetic code actually to predict for every single type of amino acid, what is the given mutation rate for histidine in this first example with EGFR in dark green here, for a histidine to change into a tyrosine or a histidine to turn into tryptophan. That never happens because there's no genetic code that permits a single nucleotide mutation to do that. So then you can make this prediction for if everything was just neutral, if we were just spraying things with mutations and they made no difference into the tumor growth or appearance of tumors, what would the distribution of mutations look like? And this is 3 demonstrations of short pieces of each protein, EGFR, KRAS, and catenin B, showing you that there's quite a distribution of how many mutations we expect to appear in different locations. Red is high, gray is low. So those mutation rates then give us a prediction. But when we look at reality, the reality is we see a very different distribution. We see very specific sites at least in driver genes that are mutated at much higher frequencies in tumors than would be expected just looking at the trinucleotide and gene mutation rates. And that tells us that these are actually selected. And basically the ratio, it's a little more complicated than this, but approximately the ratio of how often you see them here in the reality to how often you see them in the prediction tells you what the effect size is, or in other words, how much that mutation actually increases the survival and reproduction of the cancer cell lineage. In other words, how much it causes cancer. So that quantification enables us to better understand the major drivers in different genes. That's all I'm going to say about this for now, except to just comment that this is work that's already published. What we're doing now is looking at stage-specific rates so you can understand, is a therapy going to be useful early in cancer? Is it going to be late in cancer? We can make all kinds of predictions about what therapies we can develop and what will be effective treatments based on this kind of understanding of what the selective impact of every mutation is in cancer. And I'll just very quickly mention that another side of my lab, just because we just got a whole lot of funding to do it, so there's plenty of funding if someone is interested in this kind of stuff, which is we also do this fungal biology, also related to phylogenetics. So we also do multi-sample analyses with cancer, looking at multiple samples and understanding this selective effects. And then sort of in parallel, we're also looking at host invasion by fungal spores. So we look at different, very different research project, but we're looking at different fungal species in collaboration with multiple people, looking at the gene expression in those fungal species, reconstructing ancestral gene expression, and then evaluating whether or not given genes, again, what we're looking at is what's the key gene here? What key genes actually play roles in their ability to be pathogens on different fungal hosts. So here we've got entomopathogens, mycopathogens, and plant pathogens, and then Neurospora crassa, which we work on in my lab, which is a model organism. And then we are also, that's out of line. Well, I don't have the slide here, but the other thing I want to just mention is we have a very similar study that just got funded from NIH, where we're looking not at these kinds of pathogens, but at pathogens of humans. Aspergillus, which infects individuals who have bone marrow transfers. We look at skin pathogens, various fungal pathogens that we're doing. And in the lab, we don't actually work with any high-level pathogens that are dangerous to humans. We work on the model organisms, and we have collaborators who work on the pathogens. But we are sort of the clearinghouse for all the data. It all comes in, and then we analyze the evolutionary differences among these different species. And by culturing them in a common medium, we can ask lots of really interesting questions about pathogenesis and its evolution. All right, so that's all I'm going to say at the moment, unless anyone has